Welcome to the Deep Dive. We're the podcast where we really jump into complex topics, try to cut through the noise, and give you the most useful bits of knowledge. Today, we're exploring a world that's genuinely everywhere. I mean, all around us, on us, even inside us. But it stays surprisingly, mysterious and often misunderstood. We're talking about the massive, ancient, and sometimes, let's be honest, slightly alarming world of fungi. Now, when you hear fungi, maybe you just think of mushrooms on pizza or that, fuzzy stuff on old bread. But these organisms, they are so much more. They're incredibly diverse, they're everywhere, and they're absolutely critical to life on Earth. They do everything from decomposition to forming vital partnerships, and sometimes they impact our health in really profound ways. So our mission today is to give you a real shortcut to getting well informed on how fungi are classified, explore the different ways they can affect human health, and then we're going to zoom right in on one particularly unique and frankly intriguing condition called rhinosporidiosis. We've pulled together insights from a really comprehensive guide on fungal classifications and the diseases they cause, plus a specialized look at rhinosporidiosis itself. So get ready for some genuinely surprising facts and maybe some real aha moments. It should shed some light on why understanding these organisms is so crucial, not just for scientists, but really for anyone curious about the hidden web of life, including what's happening microscopically inside our own bodies.
Absolutely. And what's really illuminating when you dig into it is realizing just how much these organisms have shaped life as we know it, often silently. They've been evolving alongside us for, billions of years. Their story is deeply woven into ours, much more than most people probably imagine.
Okay, so let's start right at the beginning. We talk about fungi, we see them. But what are they really? Are they plants, some kind of super evolved bacteria? What actually makes a fungus a fungus?
That's the perfect place to start because it hits on a really foundational distinction in biology. Fungi are eukaryotic microorganisms.
Okay, eukaryotic. Break that down for us. What does that mean in simple terms?
Right, so eukaryotic is a critical term. It means their cells have a true nucleus, a dedicated membrane-bound compartment where all their genetic material, their DNA, is neatly packaged up and protected. And they also have other complex internal structures, things like mitochondria, and powerhouses of the cell, generating energy, and a pretty sophisticated system of internal membranes doing various specialized jobs. Now, this is fundamentally different from, say, bacteria. Bacteria are prokaryotic.
Prokaryotic meaning simpler.
Much simpler, yeah. They lack that true nucleus and all those complex internal organelles. Think of it maybe like comparing a highly organized factory with specialized rooms and assembly lines to just a simple open workshop. That eukaryotic cell design allows for a much, much greater level of complexity and specialization.
Okay, so they're not simple at all, really. They're quite intricate internally, more like animal or plant cells in that basic structure. That's a big difference. And I think I remember something about their cell walls being unique, too, compared to bacteria. Does that difference matter in the real world?
Oh, it absolutely does. And it's a difference with really profound medical significance. While both fungi and bacteria have rigid cell walls for support and protection, what they're made of is vastly different. Fungal cell walls are pretty robust. They're primarily made of chitin.
Wait, isn't that what you find in, insect shells?
Exactly. The very same tough, fibrous substance you find in the exoskeletons of insects, crabs, lobsters, that kind of thing.
Mm-hmm.
Along with chitin, they also have a type of lipid called ergosterol. Bacterial cell walls, on the other hand, they're made of something called peptidoglycan.
Peptidoglycan, okay.
And this difference in makeup is incredibly important when it comes to treatment. It explains exactly why many common antibiotics for bacterial infections like penicillin or amoxicillin are completely useless against fungal infections.
Oh, okay.
Those antibiotics are designed specifically to target and mess up the peptidoglycan in bacterial cell walls. It's like trying to unlock a door with a key for a totally different lock. It just won't work because the target isn't there.
So you need completely different drugs.
Precisely. We need entirely different classes of drugs called antifungals to tackle fungal infections effectively.
That's a really key aha moment right there. The very thing that makes bacteria vulnerable isn't even in fungi. So, the whole different treatment approach. And what about how they reproduce? I always think of spores with fungi, but don't bacteria have spores too? What's the main difference in purpose?
You're right. Both can form spores, but their primary functions are pretty distinct. Fungi mainly use spores for reproduction and dissemination, spreading out. Think of a mushroom releasing millions of tiny spores into the air. That's their main strategy for spreading, colonizing new places, multiplying. These spores get carried by wind, water, even animals. which is why fungi are just so pervasive.
Okay, spreading life.
Yeah. Whereas for bacteria, while they do form spores, these are often survival mechanisms for when conditions get really tough. Bacterial spores, like endospores, are dormant, highly resistant forms. They let the bacterium survive extreme heat, radiation, lack of food, just waiting until things get better.
So more about enduring hardship.
Exactly. More about enduring than reproducing in that context. So fungal spores are mostly about spreading life. Bacterial spores often about preserving it through tough times.
That makes a lot of sense. So they're complex inside, unique cell walls with big medical implications, and a distinct way of spreading. Given all that diversity in their ancient history, how do scientists even start to sort them all out? Are there established systems for classifying fungi?
Well, like with any huge group of living things, fungi can be classified in multiple ways. It helps us understand their relationships, their biology, and, their potential impact. We mainly use two big approaches. Taxonomic classification, which is about placing them in the big tree of life based on evolutionary connections, and morphological classification, which is more about what they actually look like and how they grow. Both are really essential, especially in medicine, for identification.
Right. Let's tackle the big picture first then. Taxonomically, where do fungi fit in the whole living world?
Okay, at the very broadest level, all life is categorized into three domains: archaea, bacteria, and eukarya. Fungi definitely fall under eukarya.
Along with animals and plants.
And protists, yeah. Historically, for a long time, life was just split into two kingdoms, plants and animals. Super simple. But as we learn more, especially with microscopes and genetics, that view expanded a lot. We now commonly use a five-kingdom system: Monera, Protista, Plantae, Fungi, and Animalia.
And that was a big deal for fungi.
Huge deal. It officially recognized fungi as their own distinct kingdom, separate from both plants and animals.
So definitely not plants. I've heard them confused with algae sometimes, maybe because some look a bit plant-like, but they're different there too, right?
Oh yes, very different. And this is another key point. While fungi and algae were once sort of lumped together in an old group called Phallophyta because of their simple structures, the fundamental difference is massive. Fungi completely lack chlorophyll.
Chlorophyll. No green stuff.
Exactly. Which means they cannot perform photosynthesis. That's the process plants and algae use to turn sunlight into energy. Fungi are heterotrophs.
Meaning they have to eat something else.
Essentially, yes. They have to get their nutrients from external organic sources. They usually do this by secreting enzymes onto their food source, like dead wood or soil or even us, and then absorbing the broken down molecules. This affects everything, like where they live, often dark, moist places with lots of organic stuff, and how they interact with other organisms. They can be decomposers, partners, or sometimes parasites. So despite any surface similarities, their basic way of getting energy puts them worlds apart from plants and algae.
That's a really fundamental difference, not making their own food. Okay, so within this distinct kingdom fungi, how do scientists break it down further? What are the main groups based on those evolutionary relationships?
Well, the most common taxonomic divisions, especially the ones relevant to medicine, are based on how they form their sexual spores. This gives us four main classes, each with defining features. First, we have the Zygomycetes. These are often thought of as lower fungi, maybe because they have some more primitive characteristics. A key identifier for them is their non-septate hyphae.
Okay, let's unpack this. Non-septate hyphae sounds pretty technical, but it's actually a really interesting characteristic of how they grow, right? What does it really mean for their structure?
Yeah, it's a great point to clarify. So imagine a fungal hypha that's the basic thread-like structure of a filamentous fungus, like a mold.
Okay.
In most fungi, these threads are divided into individual cell-like compartments by internal cross walls. Those walls are called septa. Septa have little pores, so stuff can move between compartments, but they still divide things up.
Right, like rooms in a corridor.
Kind of, yeah. But non-septate means Zygomycetes don't have these cross walls. Their hyphae are essentially continuous multinucleated tubes of cytoplasm, like one giant long cell with lots of nuclei floating around inside.
Wow. Okay, so no dividing walls.
Right. It looks more primitive under the microscope, and it allows for really fast movement of cytoplasm and nutrients throughout the whole structure. They reproduce mainly by making asexual spores called sporangiospores, which are held inside a little sac called a sporangium at the tip of a specialized stalk. Common examples you probably know are things like bread molds, rhizopus, and other environmental molds like Obsidia and Mucor. You find them on decaying stuff everywhere.
So a more open plan fungus structure, you could say. Sounds efficient for growth. What's the next big class after these Zygomycetes?
Next up are the Ascomycetes. Now, this is an incredibly broad and diverse class, probably the largest fungal phylum, actually. It covers a huge range of forms, everything from simple single-celled yeasts to complex cup fungi and lots of common molds. Their defining feature, the thing that links them all taxonomically, is how they form their sexual spores. These are called ascospores, and they're formed inside a special sac-like structure called an ascus.
A spore sac.
Exactly. What's truly remarkable here is just the sheer variety and ecological importance within this group, all sharing this unique reproductive method. This class includes many fungi that are incredibly useful to us, like Penicillium species.
Penicillin.
The very one, yeah. And various Saccharomyces species. Those are the yeasts we use for brewing beer, making wine, baking bread.
Helpful ones.
Many are. But this group also includes major plant pathogens and some significant human pathogens, too. Their adaptability, being able to be simple yeasts or complex multicellular forms, just makes them incredibly successful everywhere on Earth.
All right, so they're the ones with the little spore sacs, a truly versatile bunch, from medicine to baking. What about the third class?
Then we have the Basidiomycetes. These are probably the fungi you're most familiar with visually. This class includes the vast majority of common mushrooms, toadstools, puffballs, shelf fungi, the stuff you see in the woods or, by the grocery store. They form their sexual spores, called basidiospores, on a characteristic club-shaped structure that's the basidium, or base. These basidia are often lined up on the gills or pores underneath a mushroom cap.
Okay, the classic mushroom form.
Mostly, yes. But beyond the edible or just interesting-looking ones, there's a really crucial medical link here that might surprise you. A key example is Phyllobacidiella neoformans. Now, that name might not ring a bell, but it's the perfect stage, meaning the sexual spore-forming stage of Cryptococcus neoformans.
Ah, Cryptococcus. I've heard of that one. Causes meningitis.
Exactly. Cryptococcus neoformans is a very significant human pathogen, especially dangerous for people with weakened immune systems. It causes serious infections like cryptococcal meningitis, which can be fatal. So what often looks like a harmless, maybe even tasty mushroom relative can actually have a very serious side in its microscopic yeast-like form.
That's a huge leap from something on a pizza to a serious human pathogen.
Wow. And that brings us to the last group, which sounds a bit mysterious, imperfect fungi. What does that mean? And why are they so important to us?
Indeed, the last class is the Deuteromycetes. They're also widely known as the fungi imperfecti. Now that imperfect label sounds a bit negative, doesn't it? But it simply means that for fungi put in this class, scientists haven't identified a sexual reproductive state yet. Or maybe they lost it over evolutionary time. They're mainly known for their asexual reproduction, often using spores called conidia. Here's where it gets really interesting, and maybe a bit frustrating for researchers. The fungi that cause us the most trouble clinically, the ones we encounter most often as infections, are frequently the ones we know the least about in terms of their full life cycle. It's almost like they're holding out on us, hiding part of their biology. So what's the practical impact of this imperfection?
You've absolutely nailed the critical point. Despite this imperfect label, most medically important fungi actually fall into this class. Think of major human pathogens like Coccidioides mitis.
Valley fever, right?
That's the one. A dimorphic fungus causing serious respiratory and systemic infections in endemic areas. Or Candida albicans.
Oh yeah, yeast infections.
Sure.
The rush.
Very common. Ubiquitous yeast can cause everything from those superficial infections to life-threatening systemic candidiasis. For these guys, our understanding of their full reproductive cycle is just incomplete. Yet their impact on human health is massive. And this lack of a known sexual stage can make things harder. Harder for diagnosis sometimes, harder for developing treatments or vaccines, because understanding the full life cycle often gives you vital clues. So we rely more heavily on what they look like morphologically and their biochemical traits to identify and classify them.
So that covers how they're sorted by their spore sex life, if you like. But what about just how they look and grow? That must be critical for identifying them in a lab, especially if someone's sick.
Absolutely. Beyond the taxonomy, fungi also show distinct morphological forms. And these are critical for identification, especially in a clinical setting. Generally, fungi show up in one of two basic structural forms, or sometimes they can switch between them. You've got the yeast form and the mold form.
Let's start with yeasts. What exactly are they and what do they look like?
Yeasts are pretty distinct. They're unicellular organisms, single, independent cells, usually spherical or ovoid, kind of like tiny balloons. They mainly reproduce asexually by budding. A smaller daughter cell just sprouts or buds off the parent cell and eventually breaks away.
Like a little clone popping off.
Pretty much. And when you grow them in the lab on culture media, they often form colonies that look creamy, soft, maybe a bit mucoid, almost like bacterial colonies sometimes, but often with a characteristic yeasty smell. A classic example is Cryptococcus neoformans, which we just talked about. That's the asexual yeast stage of a basidiomycete. And interestingly, Candida albicans, while it's generally yeast-like and buds, has this unique trick. Under certain conditions, like inside us, it can form pseudohyphae.
Pseudohyphae, fake hyphae.
Kind of. They're elongated cells that look a bit like the threads of molds, but they have these constrictions where the cells join. It shows they're really just a chain of yeast cells that budded but didn't fully separate. They're not true continuous hyphae. But this ability helps make it a versatile pathogen.
And then there are molds. When I hear that, I picture something fuzzy, maybe colorful. Is that right?
Exactly. Your mental picture is spot on. Molds are filamentous fungi. That means they're multicellular, and they grow as long, thread-like structures called hyphae. These hyphae grow, branch out, tangle up, and when you get a visible mass of them, that's called a mycelium. That mycelium is the fuzzy, often cottony or velvety, frequently colorful mold colony you see growing on bread or cheese or damp walls.
Okay.
Under the microscope, these hyphae can either be septate, meaning they have those internal cross walls we talked about, dividing them into compartments.
Like the zygomycetes don't have.
Right. Or they can be non-septate, like in the zygomyces, where they're just continuous open tubes, common examples. Aspergillus species, you find those everywhere, decaying stuff, even in buildings.
Yeah.
Penicillium species, again, not just for antibiotics, they spoil food too. Rhizopus, the common bread mold. And that group called dermatophytes, which cause many skin and nail infections. The distinct look of the mold colonies in their hyphae are key for lab identification.
So we've got these two main forms, single-celled yeasts and thread-like molds. But what about those fungi that seem to break the rules? The ones that can be both, depending on where they are.
Ah, yes. Those are the real shapeshifters of the fungal world.
Yeah.
The dimorphic fungi. These are particularly fascinating. They have this remarkable ability to exist in both the hyphal or mycelial form like a mold and the yeast form, or sometimes other unique structures depending entirely on environmental conditions, especially temperature.
Wow, shapeshifters. That's a good term.
Really fits. It's an incredible adaptation that speaks volumes about their evolutionary success. What's truly astonishing is how precisely they control their form based on where they are. In their natural habitat, like soil or decaying plants. And when you grow them in the lab at cooler, room temperature, say around 22 to 25 degrees Celsius, they grow as that multicellular hyphal mold form.
Okay, mold in the cold.
Right. But when these same fungi infect a warm-blooded host, like us, or if you culture them in the lab at body temperature around 37 degrees Celsius, they transform. They switch to a yeast form, or sometimes other specialized structures like spherules. These are single-celled, and much better adapted for surviving and spreading within host tissues.
That raises an important question then. Why? Why would a fungus evolve this ability to change forms so dramatically?
It speaks directly to their incredible adaptability and, frankly, their virulence, their ability to cause disease. The yeast form, being single-celled and often smaller, is generally better suited for spreading through the bloodstream, maybe hiding from the immune system, or surviving inside our immune cells. Very. Whereas the mycelial form is optimized for growth, grabbing nutrients, and surviving out in the environment outside of a host. Examples of these clinically really important dimorphic fungi include Histoplasma capsulatum that causes histoplasmosis, Blastomyces dermatitis, responsible for blastomycosis, and Coccidioides immitis, the cause of coccidioidomycosis or valley fever. This ability to switch forms is a key part of their pathogenicity, their power to cause those serious deep-seated infections. It's a prime example of their sophisticated dance with their environment and their hosts.
That's an incredible overview of what fungi are, their unique cell biology, and how they're sorted out. Now let's shift focus to what happens when these organisms interact with us in a, well, less friendly way. Let's talk about the wide spectrum of fungal infections, which are called mycoses. What are some general characteristics of these infections? How do they compare to, say, bacterial infections?
Generally speaking, fungal infections, or mycoses, are often known for being chronic and slowly progressive. Unlike maybe a bacterial infection that hits you hard and fast, and then hopefully resolves quickly with antibiotics, fungal infections tend to linger. They can develop gradually, sometimes over weeks, months, even years, slowly getting worse if they're not treated. This chronicity can actually make them hard to diagnose early on.
Slow burn.
Often, yes. But, and this is absolutely crucial, while many are chronic, they can also be acute and rapidly life-threatening, especially in people whose immune systems are compromised. The severity and how quickly it progresses really depends on the specific fungus involved, how much exposure someone had, and most importantly, the person's underlying health and immune status. To help make sense of this broad spectrum, we usually classify mycoses into 5 major groups. This classification is mainly based on the tissues in the body they primarily affect and how the infection is typically picked up.
Okay, 5 major categories. Let's start with what sounds like the mildest, maybe the most common one, superficial mycoses.
That's right. Superficial mycoses are, just like the name suggests, the least invasive type. These infections are strictly limited to the very outermost layers of the skin. We're talking the stratum corneum, that dead outer layer plus the hair shaft, and sometimes certain mucosal surfaces, like the inside of your mouth or the vaginal lining. They generally aren't life-threatening. They mostly cause cosmetic issues, although they can definitely be irritating, itchy, or just, unsightly. Think of it more as a surface colonization rather than a deep invasion.
So what does this all mean for you, the listener? Many people might have actually had one of these without even realizing it was a fungal infection. Maybe they just thought it was dry skin or some kind of rash. Can you give us some common examples?
Absolutely. A very common one lots of people might recognize is pityriasis versicolor. It's also widely known as tinea versicolor. This is caused by Malassezia furfur, which is a yeast that's actually a normal part of our skin flora. But when conditions are right, maybe it's hot and humid, or you're sweating a lot, it can overgrow. This leads to discolored or depigmented patches on the skin.
Oh, those little spots you sometimes see on people's backs or chests.
Exactly. Often on the chest, back, neck, or arms. Especially in areas with lots of oil glands. The patches might be lighter or darker than the surrounding skin, so they become quite noticeable, particularly after you've been in the sun. Another example is tinea nigra. This one's caused by Exophiala warnecki. It results in dark brown or even black flat spots, usually on the palms of the hands or soles of the feet. The dark color might look worrying, but again, it's just on the very outer skin layer and usually doesn't cause any symptoms.
Okay.
Then you have hair infections like black piedra and white piedra. These are caused by Piedria horte and Trichosporin bergeli, respectively. They show up as small, hard nodules stuck along the hair shaft. They don't usually cause hair loss, but they can make the hair feel kind of gritty. And finally, a very common one, mucosal candidiasis. This is a yeast infection of the mucous membranes, most often caused by Candida albicans, which, again, is a common resident in our bodies.
Like thrush.
Exactly. When it affects the mouth, it's oral thrush. You see those creamy white patches. When it affects the vaginal area, it's vulvovaginitis, a common yeast infection. These are opportunistic. They tend to happen when the normal balance of microbes gets disrupted or the immune system is a bit down.
Okay, so those are the really surface level conditions. What's the next step down in terms of how deep the fungus goes?
The next category is cutaneous mycoses. These infections go a bit deeper than the superficial ones. They involve the entire epidermis, that's the living outer layer of skin, and also its keratinized appendages, meaning the hair and nails. The fungi causing these actually invade these structures. They're actively feeding on the keratin. These are commonly caused either by a specific group of filamentous fungi called dermatophytes or sometimes by Candida species.
Dermatophytes. Tell us more about those. Are these the ones that cause ringworm and athlete's foot?
Exactly. You're thinking of the right culprits. Dermatophytes are a specialized group of filamentous fungi, mostly from genera like Microsporum, Epidermophyton, and Trichophyton. Their unique skill, what makes them pathogens here, is that they produce enzymes that can break down and use keratin. That's the main protein in your hair, nails, and outer skin layers. This is why they only infect these specific tissues.
Oh, they eat keratin.
Pretty much. The diseases they cause are collectively known as ringworm, or technically tinea infections. The name ringworm is a bit misleading. There's no actual worm. It just refers to the ring-shaped lesions some of these infections cause. And the tinea name is often followed by a Latin word for the body part. So tinea capitis is ringworm of the scalp itchy, often causes hair loss. Tinea pedis is the common athlete's foot itching, peeling, redness between the toes or on the soles. And tinea unguium, or onychomycosis, is fungal nail infection, makes nails thick, discolored, brittle. These infections usually cause a more noticeable inflammatory response than superficial ones because the fungus is actively invading living tissue.
Right, and Candida can do this too.
Yes. Just like with superficial infections, Candida species can also cause cutaneous candidiasis. This affects the skin and nails, especially in warm, moist areas or where skin rubs together. Examples include intertriginous candidiasis that's in skin folds like under the breasts, in the groin, armpits, causes red, itchy rashes. It can also cause onychomycosis, fungal nail infections similar to dermatophytes, and it's a common cause of nappy rash, diaper rash in babies because of that warm, moist environment. So we connect this to the bigger picture. The main distinction between superficial and cutaneous often boils down to how deep the fungus gets into the skin's protective layers and whether it's actively invading living cells. Superficial just colonizes the very surface. Cutaneous actively invades the living epidermis and keratin structures, leading to more inflammation symptoms and potential tissue damage.
That clarifies the depth difference really well. Okay, so what if these fungi manage to get even deeper than just the skin and nails?
That takes us to subcutaneous mycoses. Now, these are more serious infections because they involve the dermis, the layer right under the epidermis, and also the subcutaneous tissue, which is the fatty layer beneath the skin, potentially muscle, and even fascia, those connective tissue layers around muscles. These infections typically don't happen from just casual contact. They almost always result from some kind of traumatic implantation of environmental fungi directly into the skin.
Like getting poked by something.
Exactly. A thorn prick, a splinter, maybe an insect bite, any minor injury that breaks the skin while someone's working outdoors or handling contaminated soil or decaying plants, maybe barefoot or without gloves. These lead to chronic progressive diseases. They often cause significant tissue destruction and can form draining sinuses, little tunnels that leak pus. These types of infections are much more common in tropical and subtropical regions. That's where the specific fungi live in the environment, and people are maybe more likely to get those minor injuries while working closely with soil or vegetation.
Okay, so deeper, often from injury, more common in the tropics. What are some examples of these more serious, localized infections? What do they actually do to people?
One really prominent and often debilitating example is mycetoma. It's commonly called Madura foot or Madura mycosis. It's a chronic, slow-growing infection usually affecting the foot or hand, but can happen elsewhere. It's known for a classic triad of signs. First, tumefaction that's significant, often disfiguring swelling and deformity of the limb. Second, multiple draining sinuses, those channels leaking pus and discharge onto the skin. And third, granular discharge. The discharge often contains visible grains, which are actually tiny colonies of the organism. The impact can be severe, loss of function, chronic pain. Interestingly, mycetoma can be caused either by certain bacteria, filamentous ones called actinomycetes, leading to actinomycetoma, or by true molds causing new mycetoma. So that adds a diagnostic challenge.
Wow, grains coming out. That sounds quite intense. What else falls into this category?
Then there's chromoblastomycosis. This is a chronic, localized skin disease. It typically looks like crusted, warty, almost cauliflower-like lesions that slowly expand over years. It's caused by a group of fungi called demaciaceous fungi. They're notable because they're darkly pigmented, often black or brown in tissue. Examples include Fonsegiopedrusoi and Pheophora varicosa. Another significant one is sporotrichosis. This is often famously called rose gardener's disease.
Ah, yes, I've heard of that.
Right, because it's frequently picked up after a puncture wound from contaminated plant material, like a rose thorn, carrying the fungus Sporothrix schenki. It's a chronic infection of the skin and subcutaneous tissues that produces pus and granulomas, collections of immune cells trying to wall off the infection. What's really characteristic about sporotrichosis is its tendency to spread along lymphatic channels. You might see a chain of nodular lesions going up an arm or leg following the path of the lymph vessels. And this brings us directly to the specific condition we're going to focus on in more detail next. Rhinosporidiosis. This is also classified as a chronic granulomatous disease. It's uniquely characterized by forming soft, easily bleeding, polyp-like or wart-like lesions, most often in the nose, and other mucous membranes.
Okay, so we're moving from infections mostly on or just under the skin to conditions that can really mess with internal tissues and even spread systemically. What's the next, even more severe category?
The most serious category are the systemic mycoses, also known as deep mycoses. These are infections that involve the internal organ systems, lungs, brain, blood, liver, bones, you name it. These are the most severe fungal infections, and unfortunately can often be fatal if they're not diagnosed and treated quickly. The fungi causing these are generally picked up by inhaling spores from the environment, which is incredibly easy to do without even knowing it, right? Fungal spores are just everywhere in the air we breathe.
Scary thought.
It is. Once these spores get inhaled and settle in the lungs, they can start a primary infection, often looking like pneumonia or bronchitis initially. But from the lungs, these fungi have the dangerous ability to spread hematogenously.
Through the bloodstream.
Exactly, via the bloodstream, to various organs all over the body, causing widespread disease.
These are the infections that really underscore the potential dangers of fungi, especially thinking how easily we can just breathe in spores from dust or something without a clue. What are some of the major ones to be concerned about?
Indeed. And while these infections have always been around, we are seeing them more often now. Their incidence is sadly rising, particularly in immunocompromised patients in developed countries.
Yeah.
People with weakened immune systems from things like advanced HIV/AIDS, organ transplants needing immunosuppressive drugs, cancer, chemotherapy, long-term steroid use.
Right.
But it's also important to remember, in certain areas where these fungi are very common in the environment, often in developing countries, these infections can happen in otherwise healthy people too, usually after very heavy exposure to the spores. Some key examples of systemic mycoses include histoplasmosis, caused by the dimorphic fungus Histoplasma capsulatum. The first infection is often in the lungs after breathing in spores, looking like pneumonia or flu. It might clear up on its own in healthy people, but it can spread to the skin, mouth, and many internal organs, especially if immunity is low. Then there's coccidioidomycosis, caused by Coccidioides imitacis, another dimorphic fungus. This one's mostly found in semi-arid areas, southwestern U.S., Mexico, parts of Central and South America. You breathe in spores from the dust, get a lung illness ranging from mild flu-like symptoms to severe pneumonia, and it has the potential to spread widely to skin, bones, even the brain and spinal cord.
Valley fever again.
That's the one. Then blastomycosis, caused by Blastomyces dermatitis, another dimorphic fungus, common in parts of North America. It usually starts as a chronic lung infection that can mimic other things, but it notably likes to spread to the skin, bones, and the genitourinary tract, causing quite disfiguring lesions or bone pain. And a very significant one, especially in AIDS patients, is Cryptococcus neoformans. We mentioned it earlier, the encapsulated yeast. This causes cryptococcosis. It's a major cause of meningoencephalitis, severe infection of the brain, and its linings in immunocompromised people. It causes headaches, fever, neurological problems. It can also cause lung disease. And lastly, let's mention penicilliosis, caused by Penicillium marineffe. This is another dimorphic fungus, and it's a really important opportunistic pathogen, especially in AIDS patients in Southeast Asia. It can cause a disseminated disease that remarkably looks like tuberculosis, plus characteristic skin lesions. Makes diagnosis tricky, but critical.
It sounds like a common thread through many of these really serious systemic infections is a weakened immune system, which kind of naturally leads us to our final category, which seems increasingly relevant today.
That's a perfect segue to opportunistic fungal infections. These are a distinct and unfortunately growing concern. They're caused by fungi that are generally harmless to healthy people.
Yeah.
They only cause disease in individuals whose immune systems are compromised, weakened, or somehow debilitated. The number of these infections has been rising globally, and it directly correlates with the increasing number of people with impaired immunity. Think HIV patients, organ transplant recipients on immunosuppressants, cancer patients getting chemo or radiation, people with uncontrolled diabetes, even those on long courses of broad-spectrum antibiotics which mess up the body's normal microbial balance.
So this raises a really important question. Why are we seeing more of these infections now?
It speaks to how our own health advancements and medical treatments, while life-saving in many ways, have inadvertently created new vulnerabilities. It's a paradox, almost. Organ transplants save lives, right? But the necessary immunosuppressive drugs open a window for fungi that were once totally benign to become deadly pathogens. Chemotherapy kills cancer cells, but it also damages the immune system, allowing fungi that are literally everywhere in the environment to invade.
So it's a side effect of progress, in a way.
In a way, yes. It's often a direct consequence of extending and improving life in other areas of medicine. It's generally not that the fungi themselves have suddenly become more aggressive. It's more that the host's defenses have been lowered, creating an opportunity for these normally harmless organisms to cause significant disease.
Okay, so it's less about the fungus itself being super aggressive and more about the host's vulnerability giving it an opening. What are some of the most common and concerning opportunistic culprits we see?
By far the most common fungal pathogens in this category are Candida species, leading to candidiasis. We already mentioned Candida albicans causing superficial things like thrush or skin rashes.
Right.
But in a severely compromised host, Candida can cause really serious systemic infections.
Yeah.
It can affect multiple internal organs, lead to candidemia, that's fungus in the bloodstream, or even endocarditis, infection of the heart valves. It's incredibly adaptable and just seizes opportunities when the host is weak. It can even form biofilms on things like catheters. Another major group causes aspergillosis. These are from Aspergillus species like A. fumigatus and A. niger. These molds are ubiquitous saprophytes. They live on dead organic matter. And their spores, called conidia, are constantly in the air we breathe, literally everywhere.
We're breathing them in right now.
Almost certainly, yes. In healthy people, our immune system just clears them out easily, no problem. But in immunocompromised patients, these conidia can germinate in the lungs and invade tissues. This leads to a whole spectrum of problems, from allergic reactions like allergic bronchopulmonary aspergillosis to highly invasive pneumonia and widespread disseminated disease, which can be very rapidly fatal.
Wow.
Then there's zygomycosis, also known as mucormycosis or phycomycosis. These are caused by those saprophytic molds from the class Zygomycetes, mainly Mucor, Rhizopus, and Obsidia. These can cause extremely severe and rapidly progressing infections. They're particularly notorious in diabetic patients with poorly controlled blood sugar, especially ketoacidosis, or people with leukemia. They commonly cause aggressive rhinocerebral infections, rapidly invading the sinuses and potentially the brain. Devastating. They also cause severe lung and gastrointestinal infections.
Rhinocerebral nose and brain.
Yeah, exactly. Very aggressive spread. And crucially important for HIV patients is pneumocystosis, caused by Pneumocystis jirovecii. This is a unicellular fungus that primarily affects the lungs. It was historically a hallmark of AIDS and still is the most common opportunistic infection in HIV patients, causing a severe pneumonia called pneumocystis pneumonia, or PCP. It's a classic example of something that causes zero problems in healthy people, but is devastating when T-cell immunity is compromised. And we mentioned penicilliosis earlier, from Penicillium marnefae. While it's kind of an endemic systemic mycosis, it's also a major opportunistic pathogen in AIDS patients, especially in Southeast Asia, causing that severe TB-like disease. It's important to realize these categories often overlap too, right? Some systemic mycoses like cryptococcosis or histoplasmosis definitely behave opportunistically in compromised hosts. They're dual threats. Ultimately, whether a fungus causes disease is this complex interplay between its own virulence factors, the host's specific immune status, and how the infection actually gets into the body.
We have covered a huge amount of fascinating ground today. From the basics of what fungi are, their unique biology, through this whole spectrum of diseases they cause. Now let's really zoom in, take that focused deep dive into one specific, really fascinating and quite unique condition you mentioned earlier, rhinosporidiosis. What makes this particular infection stand out so much?
Indeed. As we noted, rhinosporidiosis is classified as a chronic granulomatous disease. But what really distinguishes it is its rather unusual causative agent, Rhinosporidium saberi. This organism has puzzled scientists and pathologists for a very, very long time. Its classification has actually been quite a journey. For decades, people debated, was it a fungus, a protozoan, something else entirely? Its characteristics were just so unique.
So it wasn't clear what kingdom it even belonged to.
For a long time, no. But modern molecular biology tools have now pretty firmly confirmed its fungal nature. It seems to belong to a rather unusual group that's related to aquatic fungi, which kind of fits with how people get it.
Okay, so it's a fungus, but a weird one. What makes it so unique visually, morphologically, especially when doctors are trying to figure out what it is?
What truly sets Rhinosporidium saberi apart, and what makes it so distinctive when you look at affected tissue under a microscope, is its very unusual and quite striking morphology. Unlike typical yeasts that bud or moles with their long hyphae, this organism shows up as these highly characteristic spherical structures. They're known as spherules, or more commonly sporangia. And what's really remarkable, quite visually impressive actually, is their size variation. These sporangia can be anywhere from just 10 micrometers across up to an incredible 200 micrometers in diameter.
Okay, let's really unpack this. 200 micrometers. For a single organism, that is huge. Just for perspective, a typical human hair is maybe 100 micrometers thick, so the biggest of these could be twice the width of a hair. That's practically visible to the naked eye for a single cell. And the description makes it sound almost like a fungal Russian doll, with hundreds of tiny spores nestled inside a bigger one that's fascinating.
Your fungal Russian doll analogy is absolutely perfect. It captures the unique internal structure beautifully. And it's not even the end of it. Inside each of these large, mature sporangia, you find hundreds, sometimes even thousands, of much smaller structures. These are called endospores. Each individual endospore is only about 6 to 7 micrometers across.
Tiny stores inside the big sphere.
Exactly. These endospores are the actual reproductive units. When they get released from a mature sporangium, usually it ruptures to let them out. These endospores are fully capable of growing into new, mature sporangia themselves. This continues the life cycle and keeps the infection going within the host tissue. This unique internal development in spore release is a key part of why it causes such chronic, persistent lesions.
So where does this bizarre organism actually come from? And how do people typically get infected? Does that weird morphology give us any clues about its home?
Well, Rhinosporidium seabury is mainly found in environmental sources, specifically in dust and water. And remarkably, it's thought that fish might be its natural host, living in their aquatic environments. That's a pretty unusual reservoir for something that infects humans, suggesting a really interesting ecological niche.
Fish. Wow.
Yeah. Now, the exact intricate way it gets from, say, water to a person isn't fully mapped out molecularly, but the epidemiology looking at who gets it and where strongly suggests infection happens through direct exposure to contaminated dust and especially contaminated water. It's particularly common in people who do things that bring them into close contact with these specific environments, like bathing in stagnant pools or ponds, and this is a very specific example from the literature, people who dive into rivers or streams to collect sand from the riverbed, maybe for construction work.
So direct, prolonged contact with potentially contaminated water or dust
seems to be the key. This allows the organism to get into mucous membranes, often through tiny cuts, scrapes, or irritation. The disease itself is characterized by developing these large, often quite striking, and very friable lesions. Friable means they crumble easily and bleed very readily when touched. That's a real clinical hallmark.
But they bleed easily.
Very easily. These lesions typically look like soft, reddish, polyp-like growths, or sometimes wart-like, and they slowly get bigger. Most commonly, you find them in the nose, often presenting as a nasal polyp that bleeds a lot, or on the conjunctiva of the eye, looking like a red growth there. But occasionally they can pop up in other mucous membranes and skin sites too. Places like the ears, the larynx, the voice box area, the bronchi, airways in the lungs, the penile urethra, the vagina, the rectum, and even on the skin itself, especially if there's been some trauma.
Okay. And you mentioned it's geographically concentrated.
Extremely so. The distribution is really striking. Over 90% of all reported cases worldwide come from just 3 main regions: India, Sri Lanka, and certain areas of South America. Within India, there are specific states known to be particularly endemic, meaning the disease is constantly present at high levels. These include Tamil Nadu, Andhra Pradesh, Kerala, and Madhya Pradesh. This strong geographic pattern really reinforces that environmental link, suggesting the specific conditions the organism needs to survive and be transmitted are kind of localized to those particular aquatic and dusty environments.
So if someone shows up with these unusual bleed-easily lesions, especially if they've been in one of those specific regions, how do doctors actually confirm it's rhinosporidiosis, especially given you said it's so unique and hard to grow in the lab?
You've hit on one of the main challenges with rhinosporidiosis. It is notoriously difficult to culture using conventional lab methods. You can't just take a swab or a piece of tissue, put it on a standard Petri dish, and expect it to grow like many bacteria or common fungi. It just won't.
So lab culture is out, basically.
Pretty much for routine diagnosis, yes. Which means the primary, most reliable way to diagnose rhinosporidiosis relies heavily on directly demonstrating those characteristic R. seabury sporangia within tissue sections taken from the lesion itself. This is where pathology becomes absolutely key. Histological examination is crucial. That means taking a biopsy, a small tissue sample, slicing it very thinly, staining it with specific dyes, and looking at it under a microscope.
Looking for those Russian dolls.
Exactly. When these tissue sections are stained with common stains like hematoxylin and eosin, HED, or special fungal stains like Grocut's, methenamine silver, GMS, or periodic acid Schiff, PAS, the distinctive features become really clear. The pathologist will see a thickened surface layer, hyperplastic epithelium, signs of chronic inflammation. But most importantly, they will clearly see those unique spherical sporangia in various stages of development embedded right there in the tissue. Seeing those structures with their internal endospores is considered pathognomonic, meaning it's a definitive diagnostic sign.
Yeah.
You see that it's rhinosporidiosis.
What's fascinating here, and clearly a big hurdle for researchers, is just how resistant, how recalcitrant this organism is to standard lab methods. You said it won't grow on normal media. What about trying to grow it in animals?
Generally, attempts to diagnose it by inoculating lab animals have also been unsuccessful. This extreme fussiness about its growth requirements has historically made it incredibly difficult to study its basic biology, test potential new drugs against it, or even think about developing vaccines. There was an interesting little footnote, though. There has apparently been a reported instance of someone successfully cultivating the organism in epithelial cell culture, so growing it within living host cells in a lab dish.
Ah, so maybe it needs something from living cells.
That's the implication. It hints at a very specialized parasitic nature, maybe dependent on factors or conditions only found inside a living host cell. It really makes it a unique pathogen in the fungal world. But for diagnosis, this reliance on direct observation in tissue makes it a true seeing is believing situation in pathology. It really underscores the need for skilled histopathology.
A real challenge for diagnosis and research then. Okay, so once it is diagnosed by seeing those structures in a biopsy, what are the treatment options? How do you get rid of these lesions?
Given the nature of these growths being large, crumbly, prone to bleeding, and tending to come back, the main treatments for rhinosporidiosis are primarily surgical. This usually means the surgeon carefully cuts out the lesions, excises the growths. This is often followed immediately by cauterization.
Burning the tissue.
Essentially, yes.
Mm.
Using heat or sometimes a chemical agent to burn the tissue at the base of the lesion. This helps stop the bleeding, which can be significant, and also helps destroy any remaining abnormal tissue or fungal bits right there at the site. The goal is complete removal of the polyps and thorough ablation or destruction of the base, because incomplete removal is a common reason why these things tend to recur or grow back.
Any role for medications?
Yes. In addition to surgery and cautery, chemotherapy, specifically with the drug called dapsone, has also been noted in medical literature as being a potentially useful treatment option. Dapsone is an antimicrobial drug that's often used for other chronic infections, like leprosy. It seems to have some effect against Rhinosporidium seabury, perhaps by slowing down its growth or maturation. It's often used as an add-on to surgery, especially if the lesions keep coming back or if the disease is widespread, to try and prevent new ones from forming.
Okay. Now, thinking back, you mentioned the larynx could be an occasional site, and it's this chronic granulomatous disease. Does this unique infection have any particular implications for dentistry or oral health? I mean, the mouth is right next door to the nose and throat.
That's a really important and very practical question for dental professionals. As the source information indicates, Rhinosporidium seabury primarily causes lesions in the nose, the conjunctiva of the eye, and the eye itself. And while it can occasionally show up in other places like the ears, larynx, bronchi, urethra, vagina, rectum, and skin, the sources we reviewed do not explicitly mention the oral cavity proper, meaning the mouth, tongue, gums, teeth, as a primary or even common site for these specific Rhinosporidium lesions.
So not typically found in the mouth, on the tooth, or gums?
Not typically listed as a common site, no. However, and this is the key point for dentists, its occasional occurrence in adjacent anatomical regions, like the larynx, which is part of the upper airway, and continuous with the back of the mouth, definitely suggests a potential, though indirect, implication. Therefore, a dental professional, particularly one working in or seeing patients from those endemic regions we talked about, India, Sri Lanka, South America, might encounter this condition. It could potentially be part of a differential diagnosis if they see unusual lesions in the broader head and neck area.
So if they see something weird at the back of the throat, for instance...
Exactly. If a patient presents with an atypical, friable, polyp-like, or maybe wart-like lesion in an area like the back of the throat, the tonsil area, near the nasopharynx, basically any area close to the main sites, and they have a history that might suggest exposure in an endemic region, rhinosporidiosis could be something to at least consider, even if it's not directly involving the teeth or gums.
So it's more about recognizing something unusual nearby rather than a specific dental issue itself.
Precisely. The critical distinction is that the sources don't detail specific implications for direct dental treatments, like how it might affect fillings or root canals, or direct oral signs involving the teeth or the immediate gums or tongue. This is very different from something like candidiasis or thrush, which is very clearly and frequently discussed in a direct dental context, because it commonly occurs right there in the mouth. So for dentistry, awareness of rhinosporidiosis is mainly about knowing its regional prevalence, understanding the unique look and feel of its lesions, that friability, and recognizing the potential need for a referral. If something suspicious is seen in those nearby areas, referring the patient to the right specialist, maybe an ENT, ear, nose, and throat doctor, or an oral and maxillofacial surgeon would be the appropriate step. It's really about recognizing something unusual in the neighborhood that doesn't fit typical dental problems. Hashtag outro.
Wow, what an incredible journey we've taken today through this hidden, yet absolutely everywhere, world of fungi. We started with the basics, how they're classified, their unique eukaryotic nature, those chitin cell walls setting them apart. Then we navigated that whole spectrum of mycoses. We saw how fungal infections can be anything from common, sometimes just cosmetic, superficial skin things, right up to deeper cutaneous and subcutaneous diseases causing real tissue damage. And then those really serious, potentially life-threatening systemic mycoses that can spread throughout the body, especially impacting people with weakened immunity, a growing concern in modern medicine. And finally, we plunged into the truly unique, geographically focused world of rhinosporidiosis, learning about its bizarre Russian doll structure and the real challenges it poses for diagnosis and treatment because it's just so hard to grow.
This deep dive really does highlight just how adaptable, diverse, and often quite surprising fungi are. They're a constant presence, playing vital roles ecologically as decomposers and partners, but also capable of causing this incredibly wide range of diseases in humans. It really underscores that delicate balance between us as hosts and these potential pathogens, especially as our own medical progress sometimes creates new openings, new vulnerabilities for opportunistic fungi to emerge.
So, what does this all mean for you, our listener? Well, maybe next time you spot a bit of mold on some forgotten fruit in the fridge, or perhaps you hear about an unusual infection in the news, maybe you'll think not just about what it is, but about the incredible biological diversity and ancient adaptability it represents. Perhaps you'll ponder how intricately these often overlooked organisms interact with all life, including our own, how they shape ecosystems, influence our health, and sometimes present these fascinating medical mysteries. It makes you wonder, what other hidden worlds are just waiting for a deep dive, maybe right under our very noses? Thank you for joining us on the deep dive. We hope you feel a little bit more well-informed and maybe a lot more curious about the fungi all around us.